All right, so so let me share my screen here again. And so the first paper that I shared was a review paper that I worked on with two colleagues. Is this working? Yeah. Okay. And this was really thinking about, you know, my contribution to this paper was really thinking about infectious diseases across skills. And this was a paper very much about how you can learn from community ecology to understand and control infectious diseases. And so there was a lot of focus on heterogeneity. There was a lot of focus on, you know, different skills at which you can look at infectious diseases. And then you can also think about new ways in which you can think about the study of infectious diseases and potential control measures as well. And so this figure is from that paper. And I think this figure to me kind of sums up what I find important about this whole concept and about the whole paper. So, and it's really the idea that you can study infectious diseases at a lot of different scales, right? So people may be studying immune molecules interacting with pathogens. Other people may look at infectious dynamics inside a host. Other people may look at co-infections between that pathogen and other pathogens, or look at the interactions between the microbiome or beneficial microbes in that host and pathogens. And, you know, we can think about that skill and then think about, you know, what is the connection to the next skill, which is the population. So for example, what does that interaction between an immune molecule and a pathogen mean for subsequent transmission to other individuals in that population? Or if you have a co-infecting pathogen, what does that mean to the transmission or protective effects of other individuals in that population? And that's also where heterogeneity comes in. So heterogeneity is, of course, a very important ecological concept. You know, everyone varies, everything varies out there in nature. And that's what ecology tries to make sense of. And that's also certainly a lot of your research, if the people are here, focuses on that very much. For example, when we think about vaccination or drug treatments, the whole population is not a homogeneous mix of identical individuals, right? People vary in their age, in their immune profiles, in their habits, in their treatments, in their diets, and that could have really important effects on the spread of infectious diseases. Now, the other thing, and that's especially when we think about ecology, is the community in which organisms live, right? So humans are one population, one species, but we live in communities with other species as well. And this is especially important when we think about, you know, the jump between species. We're not alone. We're not, you know, we're not separate from other species. And a lot of the pandemics we're talking about, emerging and re-emerging infectious diseases, are based on spillover from pathogens from wild populations to us. But the reverse can happen too. And that's why, you know, there is all these limitations on tourism to see gorillas and other great apes, because we know we can spread diseases to other species as well. So we're all connected. And then, of course, going from those communities, we can scale further up to regions and the globes. And really what happens in one area of the world, especially given the mobility we have now, is going to affect what happens anywhere else, right? So if there is a cross-species transfer of a virus in some part of the world, that virus could be everywhere in a matter of days, right? And I think that COVID-19 has really shown or re-emphasized that again. We were aware of that, but it really showed it again, how quickly these things can go from a potential spillover that happens in one corner of the world to a global pandemic that happened really very rapidly. And so by looking at these things and these connections, right, and the arrows in this figure really show that these processes work two ways, right? And one of the things that we said in this paper is you can start thinking about how you can use this, not just to understand infectious disease, but also how to control infectious disease. And we had two approaches that we kind of stole from invasive species ecology, and that's defensive and offensive. So with defensive, we can think about preventing establishment. So this is what ecologists think about to stop invasive species coming in. And we can think of pathogens as invasive species. We don't want them. So what can we do to stop them from coming in? And that's where you can start thinking about some novel ways that may not be on the forefront of traditional ways of thinking about disease control. So preserving biodiversity is, you know, generally viewed as something that you do as a conservation biologist. But should we think about it as infectious disease experts as well? So reducing parasite risk, for example, through intercropping or zooprophylaxis. So, and this was actually used traditionally, and now there is calls to start this again, is increasing domestic animal populations, for example, to deflect human bites from Leishmania infected sandflies. That's one example we can think of. We can also think about cross-protective vaccination. Of course, vaccination is hard enough when we think about the pathogens we have, but if we take it one step further and say, you know, what do we need to protect ourselves against? It's not just the pathogens we already have circulating in our own population, but also those that could jump from other species. And so there are efforts for dengue vaccines that are being developed to make them protective, not just against the human circulating viruses, but also the sylvatic viruses that every now and then jump to human populations. And then we can think about the within host communities and making those protective. Right, so thinking about restoring beneficial symbionts, enhancing symbiont antagonism, you know, this kind of work happens, you know, can we use symbionts to stop the spread of infectious diseases in mosquitoes. We can also think about human treatments and think about fecal transplant or restoring gut microbial communities to really stop the, you know, infection with these pathogens or letting these pathogens grow to really detrimental densities. And then on the other hand, we have these offensive strategies that prevent dispersal. So that's saying we have it here, we want to prevent it going from somewhere else, right? And so that's where heterogeneity can come in. So understanding who are the super spreaders in the population. So when we wrote this paper, Ebola was very much in the news. And so the idea of the burials that were really super spreading events, you could start thinking about sanitary burials. Of course, now we think about this in COVID as well, and think about what are some of the super spreading events, big congregations of people, you know, big festivities and social context, those could actually be important super spreading events. So really tackling those could be an important thing to stop the spread of disease. We can also think about culling or bait vaccination, so we can reduce abundance and disease in reservoirs or populations. So this happens, for example, with vaccinating foxes or domestic dogs, to reduce rabies virus prevalence in reservoir hosts and thereby actually preventing those viruses building up in those reservoirs and jumping to humans. And then there is also, when you think about scaling, you know, the geographic scaling or the really kind of the spatial scaling in a landscape. So you can think about spatial buffers. So reducing interspecific transmission. And this has been done, for example, in some places in Asia where you can create buffers between fruit trees and pigsties to really reduce the contact between fruit bats and pigs. And then, of course, there is border control, where we can think, you know, how do you stop diseases coming in? And of course, that's something we're thinking a lot about, again, in COVID and travel restrictions, but also limiting international tire trade is actually, turns out, the tire trade actually turns out to be a very important reason for the spread of infectious disease-carrying mosquitoes such as Aedes albopictus, and actually stopping that could really prevent the spread of those mosquitoes. So I'd say this, you know, writing this paper was a really fun exercise to do, and I did it with two colleagues who are both ecologists. And then this whole idea was really the basis of my thoughts for the training grant as well as the MP3 initiative. So that's just to give you a little bit of background on that whole framework in that paper. So before I move on to talk about another paper, I just wanted to open it for discussion, see if anyone has any thoughts or questions about this.
So I have one general question.
Yeah.
In your research, is the aim to consider multiple scales within the same study at the point that you're designing it, or when you're, I guess, thinking through your results, then going back and considering multiple scales? I guess what I'm really asking is it okay, an okay approach to just consider one scale at a time, or is it better to consider both, or what your general feedback is on that?
That's a great question. I'd say, I would argue that using a Crossskills approach can make your research potentially more impactful, right? So designing your studies to get at the processes that may be important at the next scale, that could be one way of thinking about it, right? So So if you think of an example of, say we're gonna change the microbiome in the guts of humans, right? And I really should let Michael Woodworth talk about this, and please feel free to chime in. But you can think of that as, I'm gonna think about the health for that individual. But you can also think about it, what will it subsequently do to the spread of those antibiotic resistant bacteria in a hospital setting or in a larger region, right? And so that could mean that when you design that study, that you, in addition to measuring health parameters for your individuals in that study, you also look at the output of potential bacterial spread. You know, so I'd say, you know, it's interesting to think about what could be the consequences of studying, you know, the process of one scale for what would happen at the next scale. And you may not be able to, you know, study everything all at once, but certainly having some idea of how you could connect those different things, I think would be worthwhile. Michael, did you want to chime in? Because I think I talked about your research here.
Sure. Yeah. No, it's very exciting. Thanks for the hat tip. No, we love these ideas and think that, you know, looking at emergent properties and interactions of organisms at multiple levels of observation is really going to be critical to addressing problems like antimicrobial resistance. And we think that microbiome therapies like fecal transplants are fundamentally ecological therapies. And so, you know, our current position is that, you know, we are doing pretty early stage trials. And so we do our best to collect as much data as possible at all of these levels of observation. So we, you know, have collaborations with healthcare groups at the hospital level. We have collaborations with population level surveillance groups funded by the CDC that monitor the catchment area of the 8-county metro area of Atlanta. But we're also collecting, you know, stool for metagenomic sequencing and isolates for genome sequencing. And then, you know, trying to do our best to look at each one of these levels for emergent properties that can explain how these microbial community dynamics can hopefully reduce antimicrobial resistance. So, so, you know, your question is a great one to think about. You know, how do you start to get at these things? When is the best time to ask the questions? And certainly it's always better to have thought of something before you start a study. Makes it easier to get the data. But it sounds like there'll be a bit more of a robust discussion of that next week.
That's great. Lance, you wanted to chime in?
Yeah, I think that that's a great question in this figure. And then I think looking back at our introductions, I think something to keep in mind with this class is everybody's got expertise in something and nobody has expertise in everything. And our, you know, our traditional doctoral training and often our research careers tend to narrow in and work in one zone or one scale. But you can be very successful doing that, and there will be people who continue to be very successful, but keeping in mind where it fits in the big picture, I think, is something we want to talk about. And people who straddle some of these scales, it's a different community you work in, a different kind of research ecology as well. You just can't be good at absolutely everything, but understanding how it fits in this is one of the directions we want to go. Really understanding how to intervene, understand how effective an intervention at one scale is going to have implications at the other scales. So I think the question, we don't have an answer for your question, but we want to ask it every time because that's what the class and the program are really about.
Great. Any other thoughts?
I was just going to bring up maybe a good example of, you know, Lance was just talking about interventions at one scale affecting another scale. And like, obviously there's been, you know, widespread interventions to slow SARS-CoV-2 from spreading. But then now recently there's been some interesting work from Brian Grenfell that showed that, like, you know, we might expect all these endemic pathogens that are not circulating as much this year because we're having all these interventions are going to potentially come back and have bigger outbreaks in the future. And, you know, like this intervention for this year is good, but then in future years, you know, I mean, it's hard to figure out what the like correct or like moral or best strategy is, but you know, there might be consequences downstream.
Yeah. Very good. Great. So I wanted to illustrate, you know, one way in which my own lab or my own work has addressed some of this cross-skill study. And this is a study that I did together with other people on drug resistance in malaria. And this is really thinking about how can you skill from within to between host dynamics. And the collaboration was done with Kumar at the CDC, and then Mary Bushman was a graduate student of mine, and then Rustam Antia, who does a lot of infectious disease modeling, was part of this project. And so just a little bit of background on this project, focused on Plasmodium falciparum, which is the most virulent of the human malaria parasites, which is a single-celled eukaryote, and you can see how it replicates in red blood cells. So here's a parasite that's replicated and about to burst out of the red blood cells, and that bursting out every 4 to 8 hours is associated with the fevers that you get in this particular disease. And it's vector-borne, so it's spread by Anopheles mosquitoes. So what that means, it's a complex life cycle, and that already means that whatever happens to the epidemiology of a disease like that, you have to think about the different hosts in which those parasites circulate. And then the topic that we were interested in is antimalarial drug resistance. And this shows one of many, many maps that you can show about this. This is where you find malaria parasites that are resistant, in this case to chloroquine, which used to be a very widely used drug against malaria, still used in some areas. And what we can see in the dark green, that's where you find chloroquine resistance. And in other places, you still find sensitive strains to the parasite. Now, what's really intriguing is when you think about resistance in malaria, drug resistance in malaria, is that, and this is not just true for chloroquine, this is true for other drugs as well, is that it's very prevalent in high transmission areas, but that's not where it first appeared. That's not where it first evolved. And this is shown here for the origins of chloroquine resistance around the world. And you see where it originated was actually in low transmission areas in South America and Asia. And then it migrated, somehow ended up in Africa, where it then really took off and became a big problem. But it didn't evolve first in Africa, which is the highest transmission area globally for this malaria parasite. So that raises the question, why does it evolve in low transmission areas and then move to high transmission areas where maybe intuitively you think high transmission, that's where we use the drugs, that's where you would expect selection to happen, and therefore really select for these resistant parasites. So, and the way we were thinking about this, and this was very much driven by my PhD work that I did at the University of Edinburgh, where I looked at within-host competition between different malaria parasites, and I used a mouse model at the time. But this was very much based on the idea that you see a lot of multiplicity of infection in malaria. So when you look at an individual patient with malaria, you often find that there's multiple strains, multiple genotypes of the parasites circulating in the blood of that individual. And so multiplicity of infection, or MOI, is defined as the number of clones or strains in a host. And what we find is that when there's more transmission, so when the transmission intensity is higher, we get more superinfection, so more mosquitoes biting, more circulating of strains. So you see more of these multiple infections in those areas. And so the leading question here was whether within-host competition can reduce transmission of resistant parasites and thereby inhibit the spread of resistance. And my own work on mouse malaria, which is shown here, this is one paper where we track different strains of malaria in mice. And so R stands for resistant, and in the solid lines is the parasite on its own, and in the dashed lines is when it's in a mixed infection with sensitive parasites. What we can see in untreated mice here on the left is that the resistant parasites are really heavily suppressed by the sensitive parasites. The dashed lines are much lower, especially during this first acute infection. But then when you drug treat those mice, then you see there is no longer suppression. The dashed line and the solid line overlap each other, and that means that the resistant strain now reaches the same densities as it would have done on its own. So very clearly there's competition in mixed strain infections in mice, but the question was, do you find similar evidence in humans? And this is the first project that Mary did, and this was based on looking at blood samples that were available at the CDC from multiple studies around the world. We focused on Africa. And so these were samples that were collected before treatment. And chloroquine is an interesting drug because resistance to it is based on one gene. So you can sequence one gene and find out if the parasites were resistant or not. And what Mary did was use quantitative real-time PCR to look at the densities of sensitive and resistant genotypes in these blood samples. And we looked at more than 1,300 samples from 3 different countries in Africa, so Ghana, Angola, and Tanzania. Basically, we started with the Ghana study, and we got really interesting results. And naturally, we were worried that they must have been an artifact. So then we did it for Angola and Tanzania to confirm the results. And we found very similar results in all cases. Now, the first thing we're looking at here is on the x-axis, we have the multiplicity of infection. This was determined by microsatellites. And on the y-axis is parasite density, so the overall density of parasites in the blood. You see there's no positive relationship. So what it essentially means is that regardless of how many parasite genotypes there are, the overall density is about the same. And that must mean that these parasites are occupying the same ecological niche, and therefore there must be competition occurring between them. And that's exactly what we find when we look specifically at quantifying the resistant and sensitive strains. So this is data for Angola, Ghana, and Tanzania. These are the chloroquine sensitive parasites on their own in light blue, in mixed infections with the resistant parasites in dark blue. And we can see in all these 3 countries, these parasites don't reach the same densities when they're together. And we see the same pattern for the resistant strains. So the resistant strains on their own in light red reach higher densities than when they're in a mixed infection with sensitive parasites. Right? So this really shows that we see that same suppression that we see in mice. We see that in humans too. There's competition between malaria parasites within the human bloodstream. And then the other thing that we can actually look at is what you see for these resistant and sensitive parasites on their own. And by and large, when there's only one of them in the bloodstream, we find that the sensitive parasites here in blue reach higher densities than the resistant ones, indicating some cost of resistance. And we did some modeling here to see what would we expect, what did we observe in these mixed infections. And we see that in all those cases, we would actually expect a higher frequency of the resistant parasites in these mixed infections than we find, suggesting that there is a cost of resistance that is exacerbated by competition within these hosts. And that cost of resistance is also apparent when you look at the resistance over time. This is in Ghana, and we see resistance really high. And then this dashed line indicates when chloroquine was retired in 2005, and then we see the resistance strains going down. So suggesting that they're being selected against once there is no longer the drug use in that population. All right, so that was really setting the scene for a study where we really try to connect within host dynamics to between host dynamics. Establishing that yes, there is competition between malaria parasites in humans, that resistant strains can be suppressed by sensitive strains. And then the question was, how does that affect the spread of drug resistance in a population? And what we did for this was make a mathematical nested model where we're really connecting the within host dynamics with the between host dynamics. So the within host dynamics, we use differential equations to look at asexual parasites, gametocytes, which are the sexual stages that are taken up by mosquitoes. And we also modeled innate and acquired immunity dynamics. And then the between host dynamics, this was essentially an agent-based model, which means that we simulated 400 humans and 12,000 mosquitoes over time, and keeping track of every individual human. So we would keep track of the dynamics of the parasites in that human, the immune system in that human, the number of gametocytes. We would keep track of those 12,000 mosquitoes. When are they born? When are they biting? When are they biting the next person? And it's only when they bite an individual human that has gametocytes that they can get infected. Then it's about 10 days before they can then infect another human. So we really keep track of all these different processes at the time to see how the parasites then spread in that population. So to show you some within host dynamics here, this is in the absence of fitness costs. What we can look at here, this is the number of parasites in the blood on the y-axis. Over time, you can see we're looking at a lot of days in these human infections. A sensitive parasite here in blue, And these dynamics are very similar to what we find in natural human infections. Some of the best examples or the best data for human malaria comes from syphilis treatment that was actually done more than 100 years ago, in particular trials where the fever associated with malaria was used to treat syphilis. And data from those patients actually is some of the best data to look at the dynamics of malaria parasite throughout an infection. And these dynamics are very similar. Here we have high cross-reactivity. So what this means is once we have that sensitive parasite and we try to introduce a resistant parasite a little bit later, that parasite just can't come in because the immunity that is there essentially stops that parasite from coming in. We can look at high cross-reactivity and then look at a different timing here. So now we wait longer to bring in that resistant parasite, 100 days. Now you have the immune system kind of shifting to more acquired immunity, less strain transcending immunity. So this parasite that comes in can actually go up a little bit, but it's still not very impressive. Can't really grow up to very high numbers and do a lot there. Now we see differences with low cross-reactivity, and this is important, right? The different malaria genotypes vary in a lot of antigenic properties. So some of them are much more similar immunologically speaking than others. And so when you have low cross-reactivity, parasites can come in this case, the resistant parasite can come in. And especially when it comes in later, you see it can really come into that infection and establish an infection from which it can transmit to mosquitoes. And then we can look at when we add a fitness cost and look at single infections, here in blue, the sensitive parasite, in red, the resistant parasite, not much difference there. But then in a mixed infection, we can see that fitness cost really having an impact on the competitive suppression of the resistant parasite. We really see that those resistant parasites in red reach much lower densities than the sensitive parasites, right? And this is in line with what we saw in those natural populations. So the width and host dynamics of the model really capture what we know from some of the work in humans and then a lot of the work that was done in mice. But we can also look at the population dynamics and actually recreate some of the epidemiological patterns that we see in natural populations. So here what I'm showing is infection prevalence as a function of transmission intensity. So this is the bites per host per day. And when that goes up, we see infection prevalence go up. Very intuitive, and certainly in line with what we see in nature. And then we can look at the frequency of mixed strain infection. Intuitively, the more infectious bites there are, the more you're going to have these mixed infections occurring. The other thing that our model recaptures is the population, is the age structure of infections. So what we know from populations is that you have the highest densities in the youngest children, and then that declines over the as the children grow up and get older. But we also know that this decline is much steeper in areas where there is a lot of transmission. And that's because there's so much exposure to malaria that they become partially immune much more quickly. And that's, again, we see that here, that steep drop off here with high transmission intensity, much slower drop off with low transmission intensity. So the model essentially does a good job in recapitulating some of these patterns that we see in human populations. Right, and so now having shown that we have the width and host dynamics really capturing what happens, the population dynamics in line with what we see in nature as well, we can then start thinking about, okay, what happens to resistant strains that we introduce in a population where we already have endemic infections of the sensitive strains. So here we're looking at no fitness cost, no or low selection. And so we have low transmission areas, we have the sensitive parasites reach equilibrium, we introduce the resistant parasite, and we see that resistant parasite really doesn't take off. In high transmission, you can hardly see it. So that's interesting. Of course, we see high transmission, there's higher prevalence of the sensitive parasites, but that resistant parasite really can't come in. Now, if we make the cross-reactivity lower, so the resistant and sensitive parasites are more dissimilar from each other, we can see that in the low transmission area, the resistant parasites can come in and they can establish. In high transmission, that doesn't happen at all, right? So this is in line with what we see when we look at the evolution of drug resistance. It's very hard for a new resistant parasite to come in a high transmission area, much easier in a low transmission area. Now, when we look at moderate selection, the pattern changes, right? So this is when we're introducing drugs, we put selection on these parasites. We have no fitness cost here though. And now we can see that the resistant parasite can come in. And this happens both in low transmission, high transmission. It happens with low cross-reactivity and high cross-reactivity. So when there is moderate selection, no fitness cost, yes, the parasite resistant can come in any population. But when you introduce that fitness cost, which we know from our own studies and other studies is actually a real thing, that resistance often comes with a cost, we see that the resistant parasite can get established in a low transmission area, but in a high transmission area, it cannot. And we see the same with low cross-reactivity. And even with low cross-reactivity, when the parasites are very dissimilar, it's still hard for that resistant parasite to get established in a population where there is high transmission. And so at that point, you really need strong selection. so now we actually went from the idea that, you know, this resistance can emerge in a population that has low transmission. And then remember what I said about how drug resistance actually arises. It arises in these low transmission areas. And then when it reaches high transmission areas, such as in Africa, it really takes off. And so that's because you have very strong selection. And we see that happening here. So yes, once you have very strong selection, that selection can overcome the fitness cost of that resistance. Now the parasite can come in, and that regardless of low or high cross-reactivity. Right, so, but really the important thing here is that, you know, you have to start thinking about the evolution of drug resistance as first emergence and then taking off, right? So it first has to arise and then it will spread. And that first arising based on this model really seems to suggest that it is much easier and much more likely to happen in low transmission areas, and that it really requires very high selection for that parasite then to take off in high transmission areas, right? And so, and this is all driven by the competition between these strains. And the reason for that is that you have these resistant strains in a high transmission area. Where do they arise? They're always going to be in an infection that already has sensitive parasites. So it's going to really be suppressed, and that's going to stop that transmission. In a low transmission area, that parasite is more likely to find itself in a single infection without competitors and actually grow up and transmit to the next host, right? And so that is really linking that interaction that happens at the within host level, and that competition can be very much driven by the immune system and the immune responses that are in place. And that then will determine the spread of those parasites at the population level. So this is really, you know, one example, and clearly close to my heart, because this is a study that I have done, I was very much interested in, but really showing how you can understand the within host dynamics and how they drive the dynamics at the population level, and even may be able to explain global patterns in infectious diseases, in this case, the prevalence of drug resistance. So, you know, scaling from within host to population dynamics, just one of many examples, but I think this kind of study really shows the value in really linking those different skills. All right, so that's really all I wanted to share there. We have a few minutes left to discuss any questions that people may have.
I was wondering what like the next steps were with this work, or where it kind of grew from here. This is like really interesting, and it was really great to hear you break it down that way. But yeah, just like how you go from here, I guess, both in terms of when you're thinking about then application, right? So what you're doing in terms of how you're advising people, and then also like next questions. Yeah,
so next questions, you know, first thing is, this is a great model, but you know, you can make a model do anything you want in some ways, right? I think what was important for us was to recreate the patterns that we see, to give some idea of realism to a model like that. But, you know, verifying it. So with longitudinal studies in different countries, and that's what we have been discussing with Kumar and other colleagues at the CDC, you know, given sample collection in longitudinal studies where we can actually see some of these things happening in real life. So look at the changing dynamics in individual hosts, look at that suppression over time, see if the resistant parasites can come in and transmit. That is an important thing that we would like to do. Ultimately, if you want to think about how could you use this kind of thing, I think that comes back to me to the idea of taking advantage of the competition that is already there to maybe slow down or stop the spread of drug resistance. In other words, if you think about this kind of within-host competition, and we mentioned beneficial symbionts before. Here we're talking about parasites, and the question is, could you use parasites to stop more harmful parasites, in this case, resistant parasites, from spreading? And in other words, could you come up with treatments that don't completely clear the infection? Because clearing the infection, what that does is you remove all the sensitive parasites. That means the resistant parasites can now grow up and really fill that ecological niche and start transmitting. Could you come up with maybe subcurative drug treatment that reduces disease symptoms so that is beneficial at the individual level for the individual patient, but still you leave enough parasites to stop the resistant strains from coming in and spreading, right? So then you're saying, can we get enough health benefits at the individual level while also stopping transmission to the population? And I think that's a very fine balance, a very difficult balance to find, but certainly something that I think that's very worth investigating and seeing if that could be possible.
Hey, Japa, just going off that, thanks. That was a fascinating presentation. And I was just thinking talking about that kind of subcurative treatment, it makes me think about seasonal malaria chemoprophylaxis, where you give, children under 5 or under 10 drugs to just keep them from getting sick the whole malaria season. And that makes a lot of sense, but I always worry, like, what happens if the program breaks down 2 years out or 5 years out or 10 years out, and then you get a whole cohort of children that have like really never developed that partial immunity.
Right.
So, also trying to wonder is there a way to still, have like kind of like subclinical infection that still, confers immunity without causing a lot of illness?
I think that's a really fascinating question. And I think that's really at the interface of all the things that we're trying to discuss during these seminars, right? Because that requires really understanding how much immunity do you get from different levels of infection and what are the benefits for the individual, but also, based on the interplay with the population, you're going to get those parasites from other individuals. You're affecting the spread. So I think it's a great question. I don't have a direct answer, but I think that's something we should try to figure out and maybe even think about, maybe as a group exercise to maybe write a review on this at some point. I know we're out of time, and so, but this was great. I really hope to see everyone back again. I think it's a really great group. I think next week, Lance is gonna do a presentation thinking about skills. What are skills? What do skills mean to you? And things.
That's correct.
Great.
And I just posted the 2 papers to read. There'll be ones I'd like you to look at them beforehand, but you'll be looking at them after that too. So you can scan through them. So we'll see you next week.
All right. Thank you, everyone. We'll see you soon.